Hello, everyone. Thank you, Eduardo, for the introduction. I hope you can hear me well and there will be no hiccups, so everything goes fine. Yes, that's the title that I have for today. I was thinking to give you a little bit more of insight what is done in a company such as Roche regarding microbiome, because I figured that would be of interest of the audience. And First, I want to start with this already outdated slide, but I just want to tell you how are we organized so you can understand the way we set up projects, basically. So this is PRED, which is where I am in Basel, which is the research department, Pharma Research and Early Development. While you also have in San Francisco GRED, which stands for Genentech Research and Early Development, and we work together, but also we have independent lines of research. But what does span across the company is the disease areas of which we are interested in, and that's oncology, neuroscience, and immunology, basically. So those are these three pillars that you see here. Let me put this here. These three pillars that you can see here, and then where and the group where I am is located is in pharmaceutical sciences, and we really span our interest spans across these disease areas, and that's how we set up the project so we can apply them to all of them. So if you want to know, we have interest into looking into microbiome into all of these disease areas. So in Alzheimer's diseases, in Parkinson's diseases, in IBD, in cancer immunotherapy, and so on. And now I would split sort of my presentation into two parts, which is to give you an idea how are things done in clinical trials in a company such as Roche. And then the second part is really the ongoing collaborations that we have that I will touch upon later. So when it comes to clinical trials, this is very much how it flows. This is a slide predominantly done by Alison Bird from Genentech, and the video that I will show you next is also done by colleagues from San Francisco, but it really is something that we're experiencing as well in Basel. So it really starts with engaging the clinical team that is developing a drug, generating their interest, and then sitting down with them and working on protocol language so that stool sampling could be included in the protocol. Developing standardized operating procedures for stool. There's a lot of logistics and processing that needs to be accounted for so we can get the samples from the patients in clinical trials. We do the sequencing with the contract research organizations. And then once we get the data, we run we put that through our computational pipelines. Hopefully we get interesting results that will then again loop into generating interest of clinical teams and engaging their participation. So that's sort of the flow. And here I also have a snapshot of a diet questionnaire that we started implementing in our clinical trials in PRED. So for the first time ever in the 130 years almost of history of Roche, we are asking people, people in clinical trials, what are they eating? It's a very short diet questionnaire just to give us an idea of particular dietary preferences of patients in clinical trials. And then this is a really big company, and another thing that we're putting emphasis on is generating all the microbiome data in a fair way. So we want to do that natively and not get into the situation where we have rusty pipes that we need to fix. Because this is the first time ever we're doing this in Roche clinical trials. And this hopefully will play.
To ensure consistency across studies, the following collection and processing pipelines have been implemented. First, in the privacy of their own homes, patients attach a paper hammock to their toilet seat. After producing a stool specimen, they will collect a small aliquot using the provided spatula and place it into the top of the tube. The tube is then sealed and shaken. The patient stores this sample at room temperature until they bring it with them to their next clinical visit. Clinical sites collect and store these patient samples in a -20°C freezer. Every quarter, they are shipped to the Biomarker Repository Management Group at Genentech in California, where they are catalogued and aliquoted for long-term storage. Once a study is complete, all samples are shipped to the CRO Diversigen for microbiome sequencing. When sequencing is complete, the data is transferred to Genentech, where it's analysed using our customised microbiome workflows. As results become available, they are regularly shared and discussed with each study team.
Okay, so hopefully that gave you an idea of how is the whole process of getting first the sample from the patients and then the data. The video is already a little bit outdated in regards to the CRO and so on, but it doesn't matter. It really follows the path within a clinical trial of the patient, then the sample, and how we get the data, and hopefully, as I said, the results. That could be interesting. Okay, so that concludes the part on the clinical side of things, and now I wanted to show you a little bit more on the projects that we have ongoing in the group in Basel. And this is very much focused on collaborations with academic partners and on some of the internal efforts that we have. And then on this slide, the main probably thing to focus to point is the subtitle there, data integration. So we are looking into microbiome in combination with different other data types from the same patients. And that's usually how we set up our projects and how we start off. And then if you remember where we're located in pharmaceutical sciences, the way we set up our projects is so they could be applied across different disease areas. And then when it comes to data integration, microbiome and metabolomics is one of the things that we're focusing on. Where we have a PhD student who is conducting his PhD studies at the University of Oxford in this collaboration with a group of Fiona Powery. Then we have an external postdoc fellow that we're funding at EMBL in Heidelberg. Then we're putting a lot of emphasis on microbiome combined with human genetics, where we have an internal postdoctoral fellow and a master's student working on these topics. Then we're looking to microbiome signatures and sequencing data, and I will touch upon that if I have a little bit of time later on. Where we're looking into data sets that are not predominantly generated for microbiome, such as single-cell RNA sequencing and spatial transcriptomics and whole genome shotgun sequencing, where we're trying to pinpoint the signatures that might be present there. Again, we have another postdoc fellow there and a master's student working on these topics. Microbiome profiling, where we have an intern who is trying to look into biosynthetic gene clusters and strain inference. And finally, we're playing around with nanopore sequencing and applying this to microbiome samples. Okay, just a little bit more into depth regarding human genetics. We're using the EBI's polygenic score catalog to start implementing the use of polygenic risk scores, and we're trying to combine those with microbiome, something of the similar kind, such as microbiome dysbiosis index. This is pretty much work in progress and development, where we're putting the two together as we believe they are independent and can jointly contribute to in a model to derive a risk prediction metric. And when it comes to metabolomics, so this is the project that I mentioned with the University of Oxford, with Fiona Powery, where Amit is the PhD student who is looking into the metabolites that could be produced by different microbiomes. So trying to do stuff that you heard even today, but I'm sure in the previous sessions of Microbiome Virtual International Forum, where we're doing in silico modeling, based on metagenomics data, trying to see what are the metabolites produced by those microbiomes, and that allows us for generating hypotheses, essentially, and patient-specific predictions. Then we can follow up in the lab in different models. Another example of integration of metabolomics and microbiome is a project that we have with Michael Zimmerman from EMBL at Heidelberg, where we're looking into systemically mapping microbial metabolism of drugs. So this is very much building on the work done in a nonprofit organization called Pistoia Alliance that established a partnership among pharma companies at EMBL, where we're trying to do these type of things. So we are trying to identify the drug metabolites produced by members of individual gut microbiota, and then we want to really pinpoint those enzymes that are responsible for those reactions. Then when we know the chemical fingerprint of the drug and the microbiome profile, we want to use machine learning approaches in order to predict what would be the microbial metabolism of that particular novel drug. And then finally, we want to go into in vivo and test for efficacy and toxicity. Okay, so I do have maybe a minute left to give you an example. So that's really much an overview of the projects. While I would love to show you a bit more on one particular project that we have advanced on, and this is the work from Stina Ansbach, who is a master's student in her master thesis with us. Also supervised by Mark Robinson, University of Zurich, where she looked into microbial signatures in host spatial and single-cell transcriptomics data. So I don't have a lot of time to go through all of this, but I will just show you that she developed a method, a computational framework called Space Microbe, and all the credits for the name go to her, which is designed to detect microbial signatures at 10x Visium spatial transcriptomics. There were a lot of challenges, is written there. In order to do that, the framework is composed of snake-make workflow on one side, and you can see those. You can find the code on the GitLab, and there you have the link to those. And then there's also an R package that allows you to do some visualization steps and have some insights. Now we apply that to our internal data set on spatial transcriptomics. I'm just going to go really quickly through that, where we see putative biomarkers of colorectal cancer in the spatial transcriptomic data sets. And interestingly, we find a spot that is infected by EBV. We looked into the transcriptomics of that, and we have an idea that that could be a chronic infection. We're following up on that. And then we also apply that to single-cell RNA sequencing data, where we have spatial single-cell RNA sequencing data from a UC ulcerative colitis trial, and also gut microbiome data. So we're combining all of those two now. And finally, I want to conclude with this slide. Hopefully, we're not going too much over time, where this is a slide from a review on human genetics, about the potential of human genetics. And I just would like to point out we're using microbiome for all of these things, essentially, and we believe that it has potential for all of that. And with that, these are all the people that were involved in these projects, and I thank you for your attention.